Okay, so the whole story started some time ago. Probably when those of you who are parents at some point heard from kids something like, "Can I eat dirt?" or "Can I lick a pole?" Right? And your answer would be no, right? But then, as a scientist, you might ask, "Okay, why not?" So it was also the question which was sort of at the beginning what we were doing in the MetaSub consortium. Uh, and the MetaSub consortium is something which was started about a decade ago in New York when Professor Mason started asking these questions, because it was the time when the NGS has appeared, so there was a possibility not only to do the isolate, cultivate, and then check what is it. Oh, it's mine. But also that you can just collect the sample and just see everything what is there. And in the first study, which they did for the New York City Metro, so in the metro system in New York, they found that for the half of the reads which were sequenced, we have completely no idea what is it, because those sequences are coming from the organisms which are not in any of the databases. And in the first study, it was appeared okay that this which we can classify appeared to be quite close to what you have on your skin, which makes sense. Yeah, you're passing in the metro system, so probably the microbiome, which is mostly coming from the passengers. But then the question is also, can we? They identify what is in New York, and the question was, okay, how it's in the other cities, and this is how the MetaSub was born. And now every year, 21st of June, we are collecting all over the world the samples in the metro system, and this brought us a lot of data, which was the first analysis was published 2 years ago. And what was found interesting from these few thousand samples is that the urban microbiome echo important features of the cities and the city life. So where the city is located, whether it's close to the sea or not, what is the elevation, what is the density, what is the population. But what is also interesting is that the antimicrobial resistant genes are widespread in the city. So, like Ed was saying, the concept whether it's everywhere, we're just not seeing it. And the thing is that probably yes, it's still Chris is saying that he would advise any new parent to roll their child on the floor of the New York subway, because he's saying that we have so many pathogens there, but they are at a very low concentration. So you can think that this is like the multi-vaccination. So we have them; they are everywhere, and you see all of them, and they are widespread. Definitely in all the cities. The cities might have different patterns. They sometimes have one or the other, so they are pretty unique. And you can use it as I say that this is like a fingerprint. But this in fact is everywhere. You don't have cities where there's simply no antimicrobial resistance. You just have it everywhere. Interestingly, because those data are from the surfaces, but in MetaSub we also collect the air, and in air you see more. And of course, the dominant is also what you see on the surface, but in addition, you have the things which are from the other sources, where you will then not see this on the surface, because then bacteria cannot survive longer. So they just keep flying and then they just disappearing. So air could be considered as a very good like air filtering. If you could do the real-time air filtering and sequencing, this would be very good surveillance approach. And what also, in fact, has been shown today already, that the other thing is that you can also track the AMR in the sewages. And in fact, during the pandemic, it appeared that the sewage is very good source, because it's giving you the overview from the whole area, so you can quite easily and quickly detect that something is appearing. So those earlier data was with the collected and analyzed in the MetaSub, but at the same time, starting from 2017, we start giving the MetaSub data here at the CAMDA, and we're asking different questions, mostly whether you can distinguish between locations and what you can use to distinguish locations, whether you can predict based on the metagenomic data analysis that the sample is coming from one or the other places. So this we have explored pretty well over the 4 years from 2017 to 2020. And yes, there is a city-specific microbiome profiles which allow classification with high accuracy, and all approaches works well. It doesn't matter whether you just focus on taxonomic, or you go into the function, or even you just use the pure KMA based. It's working very good. Over these 4 years, I think there were published as a proceedings over 20 papers in the Biology Direct and Frontiers in Genetics. However, on the global scale, this is also what was said today, that it will not work well. Because okay, everything is spreading, but this is very connected with the local features, local conditions, which is connected with the ecological niche. So what we saw that for the classification it works, but you need to have a reference. For the prediction, it doesn't work. You cannot use the simple composition multilateration. What was interesting that there was a set of the misclassification, like the Lisbon with the New York, and the Tokyo versus Auckland. But when you look then on the globe, then you will realize that they have very similar conditions. They are almost exactly the same distance from the equator, for example, and they have a very, very similar climate. So the climate is defining the ecological niches, and then we're going to the specific biomes. And what was also interesting is that one team by Haquin in 2018, they found that for selecting, predicting from where the sample is coming from, in fact, we are using the antibiotic resistance and metabolic profiles. You can see that when they build classifier just based on the KEGG functions, then it was okay, but then using the CART profiles, so the antimicrobial resistance markers, then it was better. And then when it was combined, then the results were very, very good in terms of the classification. So all those things make that we start thinking that we should explore more this potential of antimicrobial resistance into metagenomic data. And this is what brought us here to the challenge which we propose for this year. So in this challenge, we feature both the resistance profiles of clinical isolates and environmental metagenomic sequences. And what was the concept is that we provided about 400 samples from the MetaSub consortium, so those which were collected in the metro systems in 2016 and 17 in several US cities, Baltimore, Denver, Minneapolis, New York, Sacramento, San Antonio, as well to have some kind of the international context. Also the other worldwide, so Berlin, Bogota, Doha, Ilorin, Lisbon, Sao Paulo, Tokyo, Vienna, and Zurich. And the question is, okay, so please trace AMR patterns. Look what you can find there and how you understand it. And as a supplementary challenge is that we also have provided 150 isolates, markers of the resistance identified from 150 isolates from hospitals from one of those cities. And this is what Ed was saying, okay, that it is difficult to see the same information in isolates and then environment. The question is, okay, when we're not looking to the environment, which is very broad, like where you have farming, everything, but we are focusing on the city, maybe here it will be easier. And so this was the question. And now I can say that the isolates were coming from the hospital in Baltimore. And today we will see how good people did with figuring out which city it was. So thank you and enjoy the talks about this challenge. And tomorrow we'll also have a short presentation about which direction we can go with AMR prediction and also pairing the data from the metagenomics, from the city microbiome, and from the isolates which are collected in the hospital. Thank you.